Multicellular organisms have different levels of organization, and the smallest level would be a cell. Similar cells combine to form tissues. Similar tissues combine to form organs. Organs will work together to form organ systems, and then multiple organ systems working together form the organism. Now, before things were known about cells, many individuals believed in the theory of spontaneous generation. This was widely held throughout the Middle Ages all the way through the late 1600s. And this was the belief that living things could arise from non living sources. There was even a scientific recipe to create mice, in which they said if you took dirty laundry and combined it with wheat and then let it sit for 21 days, it would create mice. Now, a scientist named Reddy in 1668 decided to refute that theory. And he disproved the idea of spontaneous generation, showing that raw meat did not generate maggots. Instead, he did an experiment that showed that maggots actually came from flies laying eggs. Because if he sealed a flask that was containing the meat, no maggots were to form. And so his experiment showed that spontaneous generation was not a viable theory. So around that time, scientists were trying to come up with explanations for how living things were put together and how they functioned. And in the mid 1600s, a scientist named Hooke examined cork under one of his very primitive microscopes and observed these box like chambers, and he named them cells. He wasn't exactly sure what he was looking at when he was looking at cork from the cork tree, but he was the one that named these chamber cells. Around that same time, another scientist named Leeuwenhoek used his microscope to observe pond water. And what he saw was organisms that were actually swimming around in that pond water. He was the first individual to see living things through a microscope. There was a span of about 200 years where not many new discoveries regarding cells were developed until we came to Schleiden in the mid 1800s. Schleiden was a botanist, and through his study of various types of plants, he concluded that no matter how diverse the plants were, they were all made of the same basic cell. Around that same time, there was a zoologist named Schwann, and he came to the same conclusion that all animals, no matter how diverse, were made of cells as well. And then in the late 1800s, a physician named Virchow would take samples from his patients and he would observe those under the microscope. He observed cells actually divide to form new cells, and he concluded that new cells must come from existing cells. So all of these findings were combined into what we today call the cell theory. And there's three parts to the cell theory. First, all living things are composed of cells. No matter how diverse the living things are, they're all composed of the same basic kinds of cell. Two, cells are the basic units of structure and function in organisms. What that says is the type of cell that is building an organism will determine the function of that organism. And finally, three, new cells come from existing cells. To observe cells, we need to use microscopes. And there's two main groups of microscopes that are most commonly used. One is called a light microscope. Light microscopes are relatively small and relatively inexpensive. Um, you can view living organisms through a light microscope. The downside is that a light microscope does not provide a great deal of detail. Another kind of microscope is an electron microscope, and these are much electron microscopes are often found in research facilities, universities. They are much more expensive. They have the ability to show extreme detail. The downside to an electron microscope, though, is that nothing that's living is able to be observed. So there's benefits and there's drawbacks to both types of microscopes. There's two main groups of cells. There's the prokaryotic cells and the eukaryotic cells. And it's important to know how these are different. While all cells are microscopic, prokaryotic cells are much smaller than eukaryotic cells. Prokaryotic cells are simpler with no nucleus. Eukaryotic cells are much more complex. They have a nucleus and they have many other parts as well. For prokaryotic cells, since there's no nucleus, the DNA is free floating in the cytoplasm, whereas eukaryotic cells, since they have that nucleus, the DNA stays within the nucleus. Prokaryotic cells make up only bacteria, where all other living things consist of eukaryotic cells: protists, fungi, plants, and animals. So it's important to understand the differences between these types of cells. And at this point, we're going to spend most of our time talking about these eukaryotic cells, the larger, more complex types of cells. And we're really going to focus on plants and animals as we talk about eukaryotic cells. Eukaryotic cells have many components inside, and those inside components are called organelles. Organelles are small structures found inside eukaryotic cells that perform specific functions. Each organelle has a specific job to do, which then allows the cell to be able to have a wider range of abilities and functions. All cells have a cell or plasma membrane, which is the outer boundary of the cell. The cell membrane follows the fluid mosaic structural model. Fluid meaning that it's flexible, it's able to move and shift, and mosaic means that it's made of different components. The main component of the cell membrane is the phospholipid. Phospholipids consist of a polar head that is hydrophilic or water loving, and two nonpolar tails, which are hydrophobic or water fearing. Since living things have a high percentage of water content, it's important that the hydrophilic parts of the phospholipid are in contact with water while the hydrophobic parts aren't. And so because of that, these phospholipids arrange themselves into a bilayer. In addition to the phospholipids, cell membranes also have a variety of proteins and carbohydrates in its structure. These function in the transport of substances into and out of the cell, enzymatic activity, where they can help speed up reactions, relay chemical messages back and forth, and cell-to-cell recognition. And cell membranes are also selectively permeable, which means they only allow certain substances to pass in and out. Outside the cell membrane, in plant cells at least, is a cell wall. And the cell wall is a rigid structure made of cellulose that gives plant cells that extra support. Animal cells have a unique part that are called centrioles. Centrioles are made of microtubules of the cytoskeleton, and their function is to organize the spindle fibers when the animal cells are dividing. Plant cells contain chloroplasts. Chloroplasts are organelles that perform photosynthesis. And they do this because they contain a green pigment called chlorophyll that has the ability to absorb sunlight and then use that to make food in the form of glucose. Now, chloroplast, this is a perfect example of where structure determines function because plant cells have chloroplasts. Plant cells are green and they have the ability to do photosynthesis. Whereas animal cells like ours, we don't possess chloroplasts. We are not green because we don't have that chlorophyll and we don't have the ability to do photosynthesis. So chloroplast, perfect example of where structure is determining then the function. All cells have cytoplasm, which is technically the space inside the cell. And it's filled with a jelly-like fluid that's called cytosol. Now, oftentimes cytoplasm and cytosol are used interchangeably to mean the fluid inside the cell, which is probably what we will use. And the cytoplasm fills the volume of the cell and suspends all the organelles in place. All cells have a cytoskeleton, which is a network of microfilaments and microtubules. Microfilaments are solid rods. Microtubules are hollow cylinders. The job of the cytoskeleton is to maintain cellular shape as well as to allow movement of substances inside the cell. Microtubules, in addition to making up the centrioles, which we've talked about already, also form cilia, which are tiny hairs that cells can use to move things across their surface. And they can also form flagella. And flagella is a tail that some cells have that they use then to swim through fluids. All cells have DNA, which is the nucleic acid, which is the genetic information of the cell. Usually, during most of the cell's life, the DNA is spread out in the form of chromatin. The only time that the DNA is able to be seen within a cell is during cell division. And during that time, the chromatin coils and condenses into the chromosomes that we know as making up our genetic information. And then once cell division is done, those chromosomes will uncoil and the chromatin will spread back out within the nucleus. Endoplasmic reticulum is a network of membranes that function in the transport of proteins and production of lipids for the cell membrane. There are two forms of endoplasmic reticulum. The rough ER is the endoplasmic reticulum, which has ribosomes on its surface, while smooth ER has no ribosomes on the surface. The Golgi apparatus is sometimes called the Golgi body or the Golgi. The Golgi prepares the cell products for storage or for export out of the cell. It does this by using vesicles. When the Golgi wants to prepare something to leave the cell, it'll put it in a vesicle, and then that vesicle has the ability to fuse with the cell membrane and leave the cell. And that's called exocytosis. We'll talk more about exocytosis and another transport method called endocytosis later in the unit. Lysosomes are organelles filled with digestive enzymes. They're more common in animal cells than they are in plants. While plants have lysosomes, they don't have as many because plant cells have a large structure called the central vacuole that has some of the same capabilities as lysosomes do. Lysosomes break down organic materials, so it has the ability to break down proteins, carbohydrates, and lipids within the cell. It can also break down old, worn out cell structures and organelles. Mitochondria is the site of energy production for the cell. It's sometimes called the powerhouse of the cell. And it does this through cellular respiration, in which it uses glucose, which is food, and produces usable energy in the form of ATP. And that ATP then is used for whatever the cell needs it to do. The nucleus is the structure that holds all of the cell's genetic information. The nucleus has a membrane on the outside called the nuclear membrane or the nuclear envelope. And so that's the outer membrane. And the nuclear envelope has holes in it called the nuclear pores. These allow things to pass in and out of the nucleus, and the nucleus is selectively permeable, just like the cell is. There's also a structure inside the nucleus called the nucleolus, and that structure makes rRNA, which is used to build ribosomes. Ribosomes, which are formed from that rRNA and protein, can either be found attached to the rough ER, or they can also be free floating in the cytoplasm. And the function of ribosomes is to produce proteins. The vacuole is the organelle of storage. In animal cells, there are very tiny vacuoles, but there's multiple vacuoles to store. In plant cells, there's one very large central vacuole, which is by far the largest organelle of the plant cell. It's much larger than even the nucleus. It functions in storage, breakdown of materials and removal of waste, like the lysosomes can do, as well as turgor pressure, which is when the plant cell has enough water in it where it is nice and firm and allows that plant to be able to stand upright. So remember, there are some organelles that are unique to just plant or just animal cells. Animal cells are the only cells that have centrioles, whereas plant cells have a cell wall, they have chloroplasts, and they have one large central vacuole. Now, there are some organelles that have special circumstances. So, talking again about the vacuole, vacuoles are found in both animal and plant cells. But remember, animal cells have tiny vacuoles and many of them, whereas plant cells only have one very large central vacuole. Lysosomes, again, both animal and plant cells have them. Animal cells have many more lysosomes, again, because of the plant central vacuole. Because of that large central vacuole, it does some of the same jobs as lysosomes do, reducing the need for as many lysosomes in the first place. And then finally, mitochondria and chloroplasts. In plants, plant cells have both chloroplasts and mitochondria. And that's because plant cells will first use their chloroplasts to make food through photosynthesis. And then they will use their mitochondria to use that food to make usable energy for itself. Animal cells only have mitochondria. And that's because animal cells can't make their own food. Animals must eat, and that's how animals get their glucose. So once they've eaten, then they use the mitochondria to create usable energy for the cell. What really makes mitochondria and chloroplasts different is that scientists believe that they developed through something called the endosymbiotic theory. And this theory states that mitochondria and chloroplast were once free living prokaryotes. They were cells of their own doing their own thing. The chloroplasts were special because they could do photosynthesis and make all their own food. Mitochondria were special because if they found food, they were able to use it to create energy. It's believed that at some point, a larger cell engulfed mitochondria and chloroplasts. It may have been doing it as a prey where it was going to eat them. It could have been that the mitochondria and chloroplasts were actually parasites that were infecting the larger cell. But whatever the reason, rather than them destroying each other, the large cell realized that there was actually a benefit to this. The large cell was now able to use the sun to make its own food and then was able to get energy out of that food. So the large cell was benefiting from what these smaller cells could do for it. And then the smaller cells were benefiting because they now had protection within another cell. The evidence that scientists have to support this idea, one, is that when chloroplasts and mitochondria are examined inside of a cell, they look like bacteria. They look like we have little prokaryotes living inside of our cells. Secondly, chloroplasts and mitochondria have their own DNA, just like bacteria do. So, because of those pieces of evidence, that helps support the idea of the endosymbiotic theory.